Alrighty, welcome back, folks. We are now getting into the nitty-gritty of DNA replication. Now, I don't know exactly what you guys are going to be doing in lab for this. Normally, we have these little foam puzzle pieces that you actually get to work with and build your DNA models and do a replication with the helicase and the DNA polymerases and all that kind of fun stuff. So I don't know what Gary and Steve are going to have in store for you, but hopefully you'll have some opportunity with the lab activities to work through these processes of DNA replication that I'll be going through here. It can be kind of a tricky process, getting the leading strand and the lagging strand and the continuous and discontinuous and Okazaki fragments and all the kind of 3', 5', all that kind of stuff. So if you're feeling challenged or you're not being able to grasp it too well, send me emails, stop by a Zoom office hour, watch some of the extra supplemental videos. There's tons of videos out there for helping you go through DNA replication. So make sure you're feeling comfortable with this whole process. It'll take a while to work through it a couple times, but the more you see it and kind of work through these steps and get some questions answered, the more comfortable you'll start feeling with it. Okay, so we set up our stage with our semi-conservative. It is going in opposite directions, and we have our enzymes of replication. So let's start identifying what we have on this screen. All right, so here we have our yellow helicase, or kind of a brownie color here. So it's not labeled. Oops, too black. So this is helicase. So we talked about helicases being the enzyme that's going in at the replication fork. It's breaking the hydrogen bonds. Here is our primer. Okay. DNA primase, RNA primase, however you want to call it, it's laying down the primer, which is a little red landing dock, right? So that was one of the rules. Our DNA polymerase can only land on a double-stranded DNA, which is, it's not truly double-stranded DNA, it's half DNA, half RNA. So, and then our DNA polymerase is this orange circle, all right? So I'm going to start on the top strand, this top strand first. Now, when we take a look at these primed ends, the 3' to 5', because these strands are complementary and because they run anti-parallel, if you have a 3' end on one spot, right, up here, it's not the color I wanted. If you have a 3' end on that spot, we know the right opposite of it has to be 5'. That's just the nature of that anti-parallel properties of those nucleotides. So if we know that this side is 5', then we know that going in this direction, this has to be the 3' end. Okay? So that's great, because what this primase is doing, it's setting down a short little primer, allowing the DNA polymerase to dock. That was our rule number 1. And rule number 2, it's setting up an exposed 3' end, which is what the DNA polymerase can read. Okay, so this kind of first picture is kind of the setup here. Now we have what's called the elongation stage, at least of the top. We're just going to be taking a look at the top strand. So here's our little piece of RNA. The DNA polymerase was able to come and land on it. And so now it's going to start adding these new nucleotides to this growing 3' end. And it knows which ones to pick because of those Chargaff's rules. If we have an A here, it's going to have to be a T here. If it is a G here, it's going to have to be a C here. So your nucleotides, those little blue kind of peg-looking T's, they're just floating around in the nucleus. You've got those because you ate food that had DNA in it. Your digestive enzymes broke up that apple DNA, and now you have free-floating A's, T's, C's, and G's of nucleotides in your nucleus, and the DNA polymerase picks the right one to match with which one it's reading. So if the DNA polymerase is reading a T on the parental strand, it will put an A in this newly forming strand, attaching it to the 3' end. It's 5' to the 3', and now we have a new exposed 3' end. You can see that right here. So that continues to happen. And if you'll notice, it's just going to chug right along, chug, 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 as the helicase continues to unwind the DNA. This is called the leading strand. Leading strand here. We can see the leading strand all the way on the top on this particular picture. the DNA polymerase adds new bases to the 3' end going towards the replication fork. So that's a really big clue. If you see DNA replication, this leading strand adding new bases, heading towards the replication fork as it's continuing to open up, this is the leading strand, and it is continuous replication because there's no break or gap in that adding of the 3' end. It's the easy one. So we start with the easy one. The other half, unfortunately, is not quite as easy because of that rule of the DNA polymerase only adding to the 3' end and the fact that DNA is anti-parallel. So let's go back up to this first setup picture, this top picture, and let's go to the bottom half. So if we have our 5' end here, what that means is your 3' has to be here. That's just the anti-parallel nature of DNA. Okay. Well, DNA polymerase can only add to the 3' end, but there's nothing here. There's nothing here for the DNA polymerase to do anything with. So where are we going to find our docking site, and where are we going to find our 3' end? Well, we have to actually go close to the replication fork on this side, where the top strand kind of started far away from the replication fork and moved towards it. In this case, our primer is right here. Here's our primer. So that's making our double-stranded little docking site for our DNA polymerase. And look at what we have right here. There's our 3' end. So in this case, the DNA polymerase is going to come in and land on that primer, which is a little red piece. And there's its exposed 3' end. So now on this lagging strand, in this second elongation part, there is our DNA polymerase. It's landed on the primer. Good. That was one of our rules. It's starting to add to the 3' end. Good. There's one of our rules. And in this case, it is moving away from the replication fork. Where the leading strand was moving towards the replication fork, the lagging strand moves away. And it will continue. It'll just continue on until it reaches, it'll just chug along until it reaches the end. That will be the 3' end. But are we done? Well, no, because what's happening at the replication fork? So if we're going to kind of draw a temporal, like a line here, even though it's not quite, I'm actually going to erase that. I don't like that idea. I changed my mind on what I wanted to draw. So what's happening at the replication fork is it's continuing to unwind, and it's opening up unreplicated DNA. Well, on the leading strand, that's not a problem because it can just keep on chugging along, working towards a replication fork. But imagine, this is the line I really wanted to draw. Let's see, I'll draw it in light green. So this primer and the very beginning of that lagging strand is actually this right here. Okay. So what about all of the stuff that happened after the DNA unwound a little bit? It's unreplicated DNA. So could we start adding to this side right here and kind of work our way towards the replication fork? No. Why? Because DNA polymerase can only add to the 3' end, and this would mean it would have to add to the 5' end, and it cannot do that. So what happens is we get what are called Okazaki fragments. And I remember when I was learning this, this was probably one of the hardest concepts, is the Okazaki fragment is as the DNA helicase is unwinding, you have to start a new primer. You have to have the primase to come in and start laying down another little docking spot. DNA polymerase is going to come in and land right here. So I'm going to try to draw this DNA polymerase. It's going to have a 3' spot to start with because that's what the primer does. And it's going to start chugging along this way, kind of like what our DNA polymerase is, until it reaches the first primer that you had done before. That DNA polymerase will pop off, and another enzyme will come in and remove that RNA primer. And then another enzyme comes in and puts a DNA base, complementary to the parent strand. And eventually you will get a completed lagging strand, but it does it in little pieces. And these pieces are called Okazaki fragments. Okay, so as the DNA continues to unwind, the leading strand chugs along. The lagging strand, right, as it's unwound, it's going to start here and it's going to go this way, but then it's continuing to unwind, so it's got to come back and go this way. I know I'm showing you stuff with my hands and you can't really see. So in the next picture, it's going to show a bigger piece of DNA replication. I think, no, that's going to be the next one. So here's that, the part of where your DNA polymerase came in, met that RNA primer, this other DNA exonuclease comes in, removes the RNA, puts in the DNA, and the little enzyme called DNA ligase seals that little nick right here. And so like I was saying, you'll get a continuous DNA strand on the leading strand, and eventually you'll get a continuous lagging strand, but it does it in these little pieces as the DNA unwinds. So here's the one I wanted to show you that's a little bit more kind of a bigger picture. So what we just kind of covered was this half. We were only covering half of this replication bubble, where the leading strand was on the top because it was heading towards a replication fork. The Okazaki fragments were kind of going like this, away from the replication fork. But because the bubble goes in both directions, excuse me, when you flip to the other side of the bubble, the leading strand here is on the bottom, and these Okazaki fragments are going to be going away. So the rules still apply. Leading strand goes towards the bubble, or sorry, towards the replication fork. The lagging strand moves away, but it's all dictated by those 3' and 5'. So here, the 3' end is on the top, going towards the replication fork, and here, on the other side, the 3' end is on the bottom, going towards the replication fork. And on the bottom of the lagging strand, the 3' end is on the bottom, going away from the replication fork. But on the other side, the 3' end is up here, going away from the replication fork. So you have, it's called bidirectionality. So you have DNA replication going in both directions at every replication bubble. And then your two bubbles will meet. So here's a replication bubble, here's a replication bubble. Eventually they'll meet, and then you get a bigger replication bubble. And then the big replication bubbles bump into other replication bubbles, and eventually you'll get two completely identical copies of DNA, barring any mutations, which is going to be another mini lesson. All right. So those are our steps of DNA replication. I know it's a lot to take in, so stop, rewind it, watch it again. Send questions, come to a Zoom office hour if you're struggling with the leading strand, lagging strand, Okazaki fragments, continuous and discontinuous replication. I know it can be really dense and heavy material, so try to get some clarification, watch some videos, other YouTube videos if that might be helpful. All right, I will see you next time. Bye.